I'm David Kelley. I'm a principal investigator at Calico Life Sciences, where we make use of machine learning to understand gene regulation, to better understand human disease and aging. And I'm helping organize the AI and molecular biology meeting for Keystone in the fall. So it's a very exciting time for computational biologists with the introduction of really powerful AI machine learning techniques into the field. I think there's a lot of optimism about what we'll be able to accomplish with the combination of really large, rich data sets paired with these methods. And there's already been some really dramatic successes, like predicting protein structure awarded the Nobel Prize. So I think it's a really exciting time to bring lots of folks together who are interested in this space to think about all of the different questions people want to ask in biology and the different types of data sets that people are able to generate, and then talk about that with the pairing of all the different methods that you can bring to bear on these types of data and questions. The Keystone meetings can be a really amazing opportunity for young scientists to talk about their research in front of a large group of leaders in the field. I'm sure there are many examples of people finding the next role that they take on in their career from contacts they make at these meetings. So the short talk deadline is May 20th. So you want to prepare those abstracts soon and get those in. And the poster deadline is later, August 21st. In the discussions I've been a part of on this topic, there's lots of interest in ways to look inside cells and see everything that's going on, to say it in a very colloquial way. And I think the technologies that enable this right now are cellular imaging with various modalities, as well as doing what I'll call spatial genomics measurements that have that spatial component so that they are kind of a type of cellular imaging. So we have a couple sessions where we're really trying to bring in major leaders in this field. The data that you get is really rich and complicated. And so the machine learning techniques that you want to bring to bear there's lots of new interesting problems for people to work on. There's lots of new development of methods. It'll be exciting to hear about that. But also just to kind of think through with the group, what are the capabilities that are going to emerge from these models? What are the new insights that we can get from peering into tissues, in particular diseased tissues, where there may be these subtle relationships that you can see with these techniques. These kind of overview highlight reel talks I find a little bit less compelling as an audience member. I would rather walk away deeply understanding one application than knowing that 10 applications are something someone was working on. And so that's a real goal of mine. And the next phase of the meeting is to really encourage the speakers to go into details in order to enhance the audience's ability to really walk away understanding some new aspect of the technology.